Our next presentation is Avid Bioservices, and I'd like to welcome Eli Hanania, Vice President, Process Development, Viral Vector Technologies, to the stage.
Thank you. I'd like to start by introducing Avid Bioservices, just to let you know that Avid has been a CDMO, pledged CDMO since 2018, but we've been producing products for more than 30 years. This gives you just a quick history about the inception and then the progression. And the key element is that in the last few years, we focused on scaling up as well as increasing capacity and capabilities. So we're really very proud of the quality that Avid brings to the table. We have more than 220 approved products in more than 90 countries. And of course, we had more than 10 PLI inspections, with the last 5 of them having 483s. We are located in Orange County, Southern California, between San Diego and Los Angeles. We have 2 campuses. The Biologics, our headquarters, 163,000 square foot, where most of the biologics, enzymes, and monoclonal antibodies are manufactured. And then the new facility in Costa Mesa, where the viral vectors are being produced. So my talk today is really going to focus a little bit on oncolytic viral vectors, and then the project that we took as an incentive for Avid to try to come up with a standardized approach that will help our clients to work with oncolytic viruses and produce the material needed. Why oncolytic viruses? As you know, most single-use gene therapy approaches are not as productive or therapeutic. And then people found out that if you try to tackle a disease or disorder by multiple approaches, you can probably get better outcome. So oncolytic viruses, of course, in addition to targeting tumors, and specifically without harming normal cells, they can also elicit an immune system. And they are large enough to have a payload, so you can really augment the impact of these viral vectors. We choose HSV1, herpes simplex virus 1, as our model. This is not new. It has been at least one of the approved product, and the virus is large enough to have a payload in it. What I'm going to show you is really what we used with the wild-type virus, and then how we really developed the upstream, downstream to manufacture this viral vector. This gives you just a quick schematic to what's involved in a typical viral vector production, starting with cell thaw and expansion. Then, of course, we infect the cells with the HSV, harvest the material, clarify it, and then go through a couple rounds of chromatography. And then at the end of the day, concentrate the virus and then diafiltrate into the final formulation, sterile filter, and that's your product. The slide shows you of the few assays that are involved in really testing each of the steps involved in that viral vector production. The 3 main assays that are used, we do genomic titer, we have ddPCR assay, and then we do infectivity assay based on TCID50, and then for the final purified material, we use MLDS, to really look at aggregation as well as the total particles. So what it involved when we started this project, upstream, it took us a couple of approaches to come up with a best way to produce the actual viral vector. It's important not just to focus purely on titer and yield, but really it's important to look at net viral productivity, factoring in the time it involves to produce that, factoring in the time it takes to do like multiple feeds, the overall cost of goods. So all that is factored in coming up with the best approach for the upstream production. Just giving you a few examples. This is, of course, with wild-type ad. We really looked at, the MOI, and then we looked at the day of harvest. As you can see, if we start with very high MOI, there is definitely a lot of cytotoxicity, so we can really, we don't get a benefit of that. If we go too low of MOI, then you might have to extend the production time, leading to more feeds and then more cost of goods. So coming up with a, 0.001 seems to be a good approach, with the net productivity being ideal. So this is just to give you an example of how we really decided on what MOI to use and what harvest days. We typically do a day 5 and a day 7 harvest. This second example is we look at different medias. I'm just focusing here on 2 medias, but we evaluated more than 6. Again, what we did here is more MOI, temperature, and media. Based on the studies we've done, looking at titers, we decided that there wasn't a big difference between these 2 medias, but the temperature really played a big role. So, doing the production at 33 was much better than, doing it at 37. The material we generated, from upstream, then we use that to really now optimize the downstream. And of course, when we optimize anything, we always consider something that a process eventually is going into GMP manufacturing. So it needs to be robust, reproducible, GMP ready, and scalable. So that's always keeping that in mind. Anything we produce small scale, eventually it has to really behave the same when we scaling up. The main approach we did, of course, chromatography steps, followed by TFF, and then final filtration. Again, also focusing on looking at step recoveries as well as overall mass balance. The downstream is more complicated because really there are several resins, several modes of production that we evaluated. And even with all of these, whenever we evaluate, we have to look at what approach we use, the capacity, and then, of course, the recovery. So since there are flow-through, there is usually involved in washes steps as well as the final peak. We had to look at all these steps and then identify how we can optimize each of these steps to really minimize any loss in the flow-through and wash, but then maximize the recovery in our peak of interest. And this gives you an example how we really picked the condition for one of the Mustang Q. We did similar studies with the Sartobind Q, and that's our second chromatography step. Again, similar things. You see some of the chromatograms are really messy, so those are not ideal conditions, but then we pick the ones that really gives us maximum recovery with minimum loss during wash and flow-through. We then repeat these studies at small scale, so we had about 700 ml of material that we generated, and then we use these steps, both Mustang Q and Chrome, and you can see that there is really hardly any losses in the flow-through as then wash, but then maximum recovery, as well as very little in the final strip. Now, does this scale up? Then we repeated these studies, now going into a larger scale. Now we repeated it at 2-liter scale, and then similarly, the same conditions that we have developed, they really behaved well in 2 liters. Now what happens if we go now larger scale? So now we repeated it at going to a 10-liter scale. And then similarly, we noticed that, yes, with the Mustang Q, we had really maximum recovery. The conditions we repeated. The Sartobind, you see there is almost 100% recovery, but that's usually not the case. It's usually, if you look at the assays and look at each of the steps involved, you can tell that, at least overall, yes, there was minimal loss in washes and then minimal loss when we did in the flow-through, but then the recovery was quite high at these steps. So this is just an example to show you, that yes, we really chose a chromatography step, evaluated the approach that based on Mustang Q, which is an IEX, and then followed by the Sartobind, and then at the end, of course, we did TFF. Again, in this table just gives you also an idea at the final and all the steps. At the end of the day, even with a 0.2 micron filtration for this virus, we are able to still get about 75% recovery. We always like to do a final 0.2 micron filtration to ensure that the product we deliver meets sterility and then is really fit for use. So I'd like then to just, give you an end with giving you why, Avid is, your choice of CDMO. We have more than 30 years of experience, both in the biologics as well as now, of course, venturing into the viral vectors. We have a very strong team that has a vast experience in all types of viral vectors. We are customer-oriented. We are not really too big of a company, but we are really moderate size, so we really know what our clients' pain points are, and then we can really work with them to meet their needs and their timelines. Of course, we are very proud of our regulatory inspections. As you saw, we are using the scaffold and infrastructure that we have in our systems, both in the mammalian as well as in the viral vectors, of course. And then, building up the state-of-the-art building, the viral vector, we did it fit from the beginning so that, the people that are involved in that building are all came from different CDMOs. So we knew what the pain points are, and then we designed and developed this viral vector and then furnished it with the state-of-the-art equipment. And then, of course, finally, we have the intimacy and the good, strong relationship with clients. So here, I just want to sum up to give you an idea, the flavor that, yes, we can handle all types of viral vectors. We focus on client needs, and then when any process that, we develop in PD, small scale, it's always intended eventually to go into a, manufacturing, clinical as well as commercial. So the facility is really ready for commercial products. And then we ensure the product from beginning, it's really scalable, GMP ready, robust, and reproducible. All the equipment that we have in PD are identical in manufacturing. All the assays we develop in analytical development are identical in QC. So there is really seamless transfer when we go from one phase into another. So I think that's mainly it. Of course, I'll be happy to answer any questions if you have any, and happy to network after that. This is my email address. I'll be around, so if you have any questions, we also have our BD team, Plamina and Kenny. So happy to answer any questions and then network and then see if you have any needs that we can help you and support you. Thank you.